All right, so we're going to look here now at something called antigen presentation. So we left off with the last part talking about receptors on various types of cells, so T cells, B cells. Now we're going to look at something called antigen presentation. So really, how do our T cells and or various types of T cells that we'll focus on specifically, so cytotoxic T cells and ones called helper T cells, how are they actually um, seeing the antigen, and how is it being shown to them in such a way that allows them to become activated and carry out their effect, right? And so this process is called antigen presentation, right? And so different cells can present antigens in different ways, right? So there will be one way on all of our nucleated cells, and then there'll be another way on what are called professional antigen presenting cells, or APCs, right? So the way that these antigens are going to be presented, and in certain cases they can be normal self antigens, or they can be foreign non-self, pathogens, right? So unhealthy antigens. So these are presented on what are called MHC complex proteins, right? And so this stands for major histocompatibility complex protein, or MHC, major histocompatibility complex. And so this is a it's a gene in our DNA that codes for these proteins, and these proteins are synthesized in the cell. They are bound to an antigen, and then that antigen MHC complex structure is then inserted into the membrane of our cells, right? And there's two main types of MHCs. There's what's called MHC1 and MHC2. Right? So MHC1 you find on all nucleated cells. Okay, all cells that have a nucleus. Right? So that includes all cells except what? Right? That includes all cells except red blood cells. Right? Because red blood cells do not have a nucleus. MHC2 you find on the nucleated cells. I'm sorry. You find on antigen presenting cells in addition to the MHC1, right? So antigen presenting cells, these are going to be cells that are specific to showing antigens to our what's called our helper T cells, right? And we'll talk about those in just a little bit. But these are found on only these professional antigen presenting cells. So these are things like dendritic cells. Our B cells are technically considered an antigen presenting cell, and a few others, right? And we'll look at some of those, right? So the way that you present an antigen will differ depending on whether it's with this MHC1 in a nucleated cell or an MHC2 in an antigen presenting cell, right? So we can look at some examples here and look at what happens whenever you present a healthy cell, right? Or whenever you present a healthy normal self antigen on a cell, right? So in a healthy cell. And then we can look at the same thing, but what happens whenever you present an abnormal or a foreign or a non-self antigen, right? So you can kind of walk through the process here, right? And so this is a endogenous protein. So this is a protein that the cell makes normally as part of its normal protein synthesis, pathway. It makes the protein. Part of the protein fragment gets added onto an MHC protein, which are these kind of orange structures here. The antigen and MHC kind of complex structure gets incorporated into the Golgi apparatus, added into a vesicle. The vesicle fuses with the membrane and inserts that MHC complex molecule with its antigen into the plasma membrane, right? And so in this case, you're presenting normal self antigens, right? So in this event that you present a normal self antigen on the outside of the cell, and our immune system comes up to it. So maybe we have a cytotoxic T cell come up to this cell with this MHC1 molecule, and it quote unquote looks at it, right? It looks at the antigen. What's going to happen, right? Well, in this case, because it's a self antigen, that cell, that immune system cell, the T cell, should ignore the self antigen, move on, and not disrupt this cell. Because in this case, this is normal. This is healthy, right? This is just part of our normal normal cellular structure to have these normal self antigens, right? So you can have something very similar to what's happening in this healthy cell, but in an unhealthy cell, right? So the basic process looks more or less the same. However, notice here, where is the antigen originally coming from, right? It's coming from outside the cell, or in this case, it's coming from a virus, right? So the virus has invaded the cell. We then break down the virus into these smaller fragments. We incorporate it again onto an MHC1 molecule. So to this point, everything is the same except that the antigen itself, right, in this case the virus, is not normal, whereas the other one had a normal self protein, right, or self antigen. So now we've incorporated this viral peptide fragment onto the MHC molecule. It gets incorporated into the Golgi, packaged into a vesicle. The vesicle gets secreted and incorporated and incorporates that MHC molecule with its, in this case, foreign antigen into the membrane, right? So in this case, now that we have a foreign antigen in the membrane attached to an MHC1 molecule, when our immune system cell comes over, right, and again, maybe it's a cytotoxic T cell that we'll talk about later, it's going to see that and it's going to view it as abnormal, non-self, and it will then exert an immune response, right? And so we'll talk about later how you actually initiate that immune response, right? How does the cell become activated, and then what is its effect after it becomes activated, right? And so this is what's happening with an MHC1 molecule, right? You also have the MHC2, and so remember the MHC1, these are on nucleated cells, right? Now, another thing I should mention, let me go back just a second. For these unhealthy cells, right, so we dock this MHC1 with a foreign antigen. In this case, the cell is essentially, the cell is going to die. Okay, the cell is going to be destroyed, right? There's no trying to save this cell. It's been infected. It is compromised. It is now a sacrifice. We're going to kill this cell so as to prevent this cell from continuing to produce these viral proteins and spread it to additional cells, right? So with this MHC1 in these nucleated cells, in this case, that are unhealthy now because they've been infected, this cell is going to be destroyed, right? Now, with our MHC2, remember these are on the surface of antigen-presenting cells, right? Those professional APCs, okay? And so you have an MHC2 molecule. These are taking exogenous antigens, so meaning antigens coming from outside the cell. So these are typically going to be foreign antigens. And then they're going to be added to our MHC2 molecule, moved to the Golgi, incorporated into the membrane, and then it's going to allow specifically our helper T cells to recognize them, right? So we can look at a very similar graphic to what we looked at with the MHC1, right? So in this case, now we have a exogenous antigen. So meaning the antigen is coming from outside the cell, in this case a bacterium, right? So that's going to be abnormal. It's going to be incorporated into, it's going to be endocytosed, right? So remember, endocytosis is the process of bringing something into the cell, and specifically this is phagocytosis, right? It's incorporated into what's called a phagosome. This phagosome fuses with a lysosome and forms a phagolysosome, right? So this lysosome contains, digestive enzymes and chemicals that are going to break down this bacteria, and then portions of that bacteria, that antigen, are going to now be added to our MHC2 molecules, right? So now we've got this foreign antigen fragment. It is now added to the MHC2 molecule, and then that whole complex is going to be added and incorporated into the membrane, right? And so again, these are specifically in our antigen-presenting cells. So dendritic cells, macrophages, B cells, right? That's what these are doing, okay? So now, the next time our immune system cell comes over and it views this, right, it's seeing this as a foreign antigen, and it's going to initiate an immune response, right? It's going to initiate an immune response. In this case, the antigen-presenting cell, you're not going to kill this cell because its job, right, its role is to present these foreign antigens, right? So you're not going to destroy it. The cell isn't infected. It intentionally brought in this bacterium, processed it, and then is now presenting it on its surface. So you're not going to destroy this cell. You would destroy the other cell that was involved with the MHC1 in the unhealthy cell, right? And so keep that in mind as we're talking about these kind of two similar but slightly different processes. Okay, and so this is showing the interaction between some of those MHC molecules and our T cells. Right now, you'll notice here. So here you can see MHC1 with an antigen. Here you can see MHC2 with an antigen. You see your T cell receptor, and you see another T cell receptor here. Now, what you also see is these other terms called CD8 and CD4, right? And you can see them here, CD4 or CD8. So what the CD stands for cluster of differentiation, right? Cluster of differentiation. So these are just additional surface markers, additional surface proteins that our T cells have specifically, right? And we'll talk about in a little bit how you end up with cells that have one or the other, right? So where this comes into play is the specific type of T cell, whether it has a CD4 marker or a CD8. That means it's going to interact with either a MHC class 1 or an MHC class 2, right? So the way you remember this is when you multiply the number associated with the CD molecule and you multiply that with the number associated with the MHC molecule, you should get 8. Okay, so example here, whenever you multiply, let me come over here. So whenever you multiply CD8, okay, when you multiply CD8 with MHC1, you should get 8, right? So 8 times 1, you get 8, right? When you multiply CD4 times 2, right, 4 times 2, you get 8, right? So when you multiply the CD number and the MHC number, you should get 8. That's telling you the interaction between these. So CD8s go with MHC1s, CD4 T cells interact with MHC2 molecules, right? So we'll get to this in a little bit. The CD8 T cells, these are called cytotoxic T cells, right? The CD4 T cells are called helper T cells, right? So helper T cells interact with MHC2, cytotoxic T cells interact with MHC1, right? Now, these MHC molecules are relevant for several things, including the donation and transplanting of organs, right? And so giving an organ to another, a heart, a liver, a lung, something like that, a kidney. And so they are donors are tested for their MHC antigens in their blood group to determine compatibility, right? And so you want individuals with a close compatibility to donate to each other because their antigens and those MHC molecules are similar to the point that their immune system, the recipient's immune system, will be able to hopefully accept them, right? Now, oftentimes, they're not going to be identical, right? So nobody has exactly the same molecules. And so there will be some degree of, immune response, right? And so oftentimes individuals who have organ transplant, they've had a heart transplant, they've had a kidney, something like that, a lung, they will take immunosuppressant drugs, right? And so this is to calm down their own immune system such that they are not attacking the donated organ that they've just received, right? And so this is beneficial in that context, but it also can maybe place them at a higher risk for infection because they are kind of decreasing and hindering their immune system, right? So we're going to stop here. We're going to pick up in our next part and begin talking about the kind of life cycle of a lymphocyte, T cells and B cells, in terms of where they're formed, how they're activated, and then what their effector response will be.